So we know prokaryotes have been around for a long time. Probably about 4 billion years or right around there, right? That's a long time. And for most of that time, they were the only forms of life on Earth. So they had the entire Earth to use, to live on, and to exist on. So that resulted in them being able to fill a bunch of different what we call niches. Niches are just a role an organism plays in its environment, how it gets its food, where it lives, things like that. There were prokaryotes that liked oxygen gas and were aerobic. There were some that didn't like it and then were anaerobic, lived in anaerobic environments. There were some prokaryotes that were great at consuming hydrogen sulfide, others that released methane gas. Some lived in cold places, some lived in warm places. There are many different types of environments that were available for prokaryotes to exist in, and they evolved to live in those different environments. And we have a lot of the descendants of those species around today still. And prokaryotes are everywhere today. We can't see them with our naked eye, but they're on every surface we touch. They're in the air, they're inside of us, they're on us, prokaryotes are everywhere. In fact, more than 75% of species on our planet that exist today are prokaryotes. That's astounding. We can't even see them. And often we get so concerned about species that we can see, like losing polar bears or the loss of the rainforest. Not saying that those aren't important, but in the grand scale of things, that's a very small percentage of total species on our planet. So the question we're asking here is, why are prokaryotes so diverse? So they've been here for a really long time. That's part of it. And that's given them the time frame needed to evolve into a bunch of different ways of life. But they have some unique characteristics that aided in that process of evolving into many different forms of life. Okay, so for example, you've probably heard of the ozone hole, right? The ozone hole is a hole in our atmosphere which increases the amount of UV light that comes through that hole. So ozone basically acts as a shield against UV light coming from our sun. So the ozone hole is always forming in the southern hemisphere. It tends to form over Antarctica, Australia, and New Zealand. And this ozone hole in our atmosphere, people that live in that area, under that ozone hole, tend to get higher rates of skin cancer during their lifetime compared to people that don't live near that ozone hole. Why? You're like, wait, we're talking about bacteria. Why are we talking about the ozone hole? Well, the ozone forms from oxygen gas in our atmosphere. And remember, oxygen gas wasn't in our atmosphere until prokaryotes put it there, until cyanobacteria started photosynthesizing. So ozone forms when it reacts with UV light to form an O3 molecule. So before cyanobacteria, before all that oxygen gas in our atmosphere, there was no ozone layer. No ozone layer means more UV light hitting our planet and hitting the surface of our planet. And UV light can damage DNA. So we would have higher rates of mutation. So more UV light hitting our planet, potentially more DNA damage happening or DNA mutations happening, which could result in more variation occurring in these organisms. And also remember that in prokaryotes, their DNA is not protected by a membrane. It has even less protection from UV light, even without an ozone layer. So we put these together, more UV light, less DNA protection. The mutation rate of early life on Earth was probably way higher than it is today. More mutation is not necessarily a bad thing. Mutations just result in new genes, new forms of a gene, right? Some mutations might be bad, some might be good. Most are going to be neutral. But the key here is that we're getting new forms of a gene, new combinations of genes that arise that could be helpful and could allow these organisms to exist in many of the different types of environments that existed on early Earth. So we talked about prokaryotes and how they go through binary fission and that they can reproduce very quickly, right? So they can make copies of themselves. But again, binary fission produces copies, it produces clones, not genetic diversity. So in organisms that go through sexual reproduction, that recombination of DNA from the two parents results in genetic diversity. That doesn't happen in binary fission. So even though there were prokaryotes going through binary fission, and we think that there was a higher rate of mutation due to DNA damage occurring from a lack of an ozone layer, that probably wasn't enough to produce enough genetic diversity to result in the huge variety of prokaryotes that we see. So there had to be another source of diversity, another source of genetic diversity introduced to these prokaryotes. So if they don't get this diversity from sexual reproduction, where does it come from? So there's other processes in prokaryotes that have led to genetic diversity in what we call horizontal gene transfer. Horizontal gene transfer is different than what we typically think of gene transfer as, like vertical gene transfer. So like passing your genes on to your child, or you got your genes from your parents, right? That's vertical gene transfer. Horizontal gene transfer is moving pieces of DNA from one individual to another. This is pretty common in prokaryotes. It happens less so in eukaryotes, but in prokaryotes, this is relatively common, this horizontal gene transfer. So we have 3 different ways that prokaryotes can exchange DNA between individuals, and that's transformation, transduction, and conjugation. Now in transformation here on the left, we see a prokaryote is picking up DNA from the environment. So in transformation, like we see here on the left, what's happening is this prokaryote is picking up DNA from the environment. In this case, there's a little free-floating piece of DNA. The bacteria picks it up, it's being absorbed by the cell, and then that plasmid ends up in the genome of that organism. And then this DNA will be passed on to the next copy of this organism that it makes once it goes through binary fission. So bacteria will pick up random pieces of DNA from the environment. And actually, we've kind of hijacked this process, and we've used it, humans have used it to our advantage. So we can introduce DNA that creates a product that's useful to us, then grow up the bacteria and isolate that product. So that's transformation. In transduction, a virus is moving DNA from one cell to another. Yeah, so prokaryotes can get viruses just like us, right? Prokaryotic viruses are called bacteriophages, and they actually enter a cell and break down the host cell's DNA, and then they build up new viral particles by wrapping a protein coat around the pieces of their viral DNA. Sometimes a piece of that bacterial DNA can be wrapped up in that viral package, and it can go on, and when the virus infects another bacteria, it can transfer some of that bacterial DNA with its viral DNA. And then that viral DNA might be incorporated into the new bacterium's genome. So this is transduction. In conjugation, this process is an active process that's controlled by the genes of the prokaryote. If the organism has the right genes and under the right environmental conditions, prokaryotes might form what's called a sex pilus between the two individual cells that attaches the two cells together and then lets them transfer DNA between that tube. So they can transfer small bits of DNA between individuals. So all of these processes act to mix up the DNA of individuals and create genetic diversity. And genetic diversity, variations in the traits an organism has, is a key component for natural selection. So many different forms of a trait might be helpful in some environments, some traits might be helpful in other environments. So putting this all together, we have high rates of mutation, we have a lot of mixing, horizontal gene transfer in prokaryotes, and a long period of time, at least 3.5 billion years, probably longer, that sets up the conditions for prokaryotes to be able to go through huge amount of diversification.